Hey everyone, so we might get started. Thank you all for joining us today. This is our second online event for Pawsey Friday, so I'm glad to see so many of you here today. My name is Aditi. I am a part of the marketing team at Pawsey, and I work to run a lot of events that we do here at the centre. So welcome to this Pawsey Friday. It's a supercomputing showcase where we're going to hear from 3 researchers using Pawsey supercomputing systems. I'd like to begin by introducing Mark Stickles. He's Pawsey's Executive Director, who's going to provide you with an update on the centre. Thanks, Mark.
Thanks, Aditi, and welcome everyone. I can see a few of my my Pawsey colleagues online, and a couple in the same building as me. I'm in my office on a rare visit to site. Most of our colleagues are working remotely and continue to do so. So great to see some familiar names of key scientists and users of Pawsey, and hopefully some perhaps from people with first-time interest in Pawsey. So welcome one and all to this event. I would like to acknowledge country to start our presentation, and acknowledge that I'm standing on, and as are some of my colleagues, the country of the Whadjuk people of the Noongar Nation, and acknowledge them as the traditional owners of the land on which we're meeting, and pay my respects to their elders past, present and emerging. And on behalf of all of us, as we've got people dialing in from all around the country, I think we can pay our collective respects to the traditional owners of all the lands of our vast nation. In terms of a bit of an update on Pawsey, we're getting to the end of the financial year, so I can't believe it's the end of June. That's just sort of approaching us this weekend and into next week. And this is, if you like, my last official activity before I actually have a week off. So I'm having some leave next week, and I offer that as just an encouragement for all of us. If you can get some time to have a break, please do so, because the year is an unusual one, and hopefully we've kept safe and well through some challenging times. Pawsey's had a tremendous few months. We've continued to progress our major projects, and we're really pleased this week to announce a partnership with the Australian Space Agency to launch the Australian Space Data Analytics Facility. So in partnership with the Australian Government, the West Australian Government, and through the WA Data Science Innovation Hub, Pawsey is going to host a new facility to really work with industry and data providers with facilities, space-based facilities, to make that data available for industry and economic application on Earth, so to speak. It's a great initiative, and we're in that sort of startup phase as we run some stakeholder consultations in July and August. So watch this space for news on that initiative. Our capital refresh is progressing at some pace. We have a great resource on our website that gives you an overview of the $70 million investment in upgrading Pawsey's infrastructure, essentially replacing the hard-working and long-serving Magnus and Galaxy infrastructure. That project is now at a really interesting point, in that we have identified our preferred vendor for the main system replacement, and we're going through the commercial in confidence negotiations to refine that decision. So in a couple of months' time, we hope to be signing that contract and moving towards implementation. But there are other components to the capital refresh. The Murchison Wide Field Array project has had a dedicated cluster purchased through that program, and we're in the process of installing that cluster at Pawsey now. We also will be releasing within the next week. We plan to release the tender on the long-term storage solution as part of our capital refresh program. So that's an important component in a very data-intensive world that we work in. I'd also like to give a shout out to my colleagues that are supporting training through Pawsey. We've got quite an extensive program of training supporting user uptake of Pawsey systems, and over the next month, we'll be doing advanced courses around our cloud infrastructure and also some introductory HPC courses in July. We'll also hold a user forum, a virtual user forum in July that encourage interested users to register for when that information is available. So a lot's happening at Pawsey, and I would like to acknowledge the tremendous contribution of my colleagues and the work that they do to support our national centre. I would, and you'll hear a bit more about that from Marco and Chris in the next phase. But I would like to just offer my personal thanks to our three expert speakers who are going to share some of their insights this afternoon around working with supercomputing to accelerate their science and the impact of that science. So we really are proud to support your work and appreciate your time to share that with the wider community. So looking forward to this afternoon's presentation. So thanks everyone, and back to you, Aditi.
That was great. Thanks, Mark. And I'm sure you didn't mean the pun when you said when you asked everyone to watch this space, but we are doing some cool things in the industry now as well. Okay, so thanks, Mark. I want to introduce you all now to Chris Harris. He's a supercomputing specialist at the Pawsey Centre. He drives the Pawsey Uptake Projects, where our three researchers have come from to use expert staff help. So Chris will give you a bit of an overview of those uptake projects now.
Thanks, Aditi. So Marco and myself are from the supercomputing applications team at Pawsey, and we help researchers develop, install, configure, and run their software on Pawsey's supercomputing infrastructure. And our uptake work can take a wide range of forms. Every researcher's project is unique, and so they can vary across a wide range from at the sort of really large end. We have formal calls for large projects where we have dedicated staff time for many months working in depth on software, all the way through to sort of more informal projects that emerge. One of the terms we have in the team is tickets of unusual size, where just as part of dealing with user support tickets, we can find there's opportunities to collaborate and really improve the way that things run on the system. In addition to the sort of uptake work we do, there's also supporting training. As Mark touched on earlier, we do develop and run our own specialist training, but we also support international specialists and experts coming and delivering training as well. Mark will be talking more about that in a little while. To find out more about our uptake projects, and so if you're interested in getting help with using our infrastructure, two great ways to do that is to send in a ticket to our user support desk just to start off an interaction. And we've also been running weekly Pawsey Ask Me Anything sessions where you have the opportunity to sort of jump into a video chat and just talk more informally with our staff about the things that can be done. And so I'll pass over, I guess, to Marco to talk a little bit more about Nextflow.
Yeah, thanks, Chris. Thanks, Aditi. Hi, everyone. So I'm Marco. I'm part of the supercomputing applications team with Chris here at Pawsey. And right now, I'd just like to introduce a bit this special uptake and training event we had last year at Pawsey. Also, an increasing number of researchers come to us with data-intensive, highly structured workflows to analyze their data. So these are often composed of they're quite fragile ecosystems, often composed of a set of softwares that need to be run, scripts to run them, and the dependencies between different tasks that need to be run in the right order to get meaningful results. So to be able to get meaningful, reproducible results in a reasonable amount of time, researchers then need a way, in our opinion, to streamline and simplify the way that these workflows are designed and then deployed in our supercomputers. And this is basically the reason behind our choice to organize a structured training event that focused on a very popular workflow tool, so a tool to manage structured workflows, that is called Nextflow. So what we did is last November, we secured an entire week here at Pawsey. We invited the two lead developers of this tool, Paolo and Ivan from the Barcelona Center for Genomic Regulation. And then we organized and structured an event that was composed of 2 days of training, followed by 2 days of hackathon straight after. And the idea was then to give an opportunity to researchers not only to learn, but also to apply what they just learned straight away on some real problem from their real research. And that's basically what happened. So the event was attended by about 35 people from all across Australia. And today we have the pleasure to host 3 of these researchers for this Pawsey Friday. So if Aditi, do you have anything to add right now?
No, if you'd like to introduce our first speaker, that would be great.
Yeah, that's perfect.
Thanks, Marco.
Sure. So our first speaker today is Mahsa Mousavi Derazmaleh. Apologies for the pronunciation. So Mahsa finished a PhD in applied bioinformatics at UWA, and she's currently a postdoctoral researcher in the Trend Lab at Curtin University. She works on a NHMRC-funded project to determine the causative agents of tick-borne disease in Australia using metabarcoding techniques. So as part of her role in Trend, she also develops bioinformatics pipelines for the analysis of environmental DNA data, which is one of the major research focuses in Trend Lab. And Mahsa will be presenting today a talk on a workflow for analysis of environmental DNA, eDNA data. So up to you, Mahsa.
Thank you, Marco, for introducing me. I'll just share my screen. Okay, so today I'll be talking about a workflow that I developed about analysis of environmental DNA and how Pawsey helped with improving this workflow. To start, I'd like to give you a quick perspective on environmental DNA. We know that when organisms interact with their environment, they leave behind traces of their DNA in their surrounding in forms of shedding cell, mucus, and feces, which accumulates before it starts to slowly disappear as the time goes on. Combining this knowledge with advances in sequencing and metabarcoding approaches enabled researchers to complement traditional methods of biomonitoring and assessment of biodiversity. Environmental DNA has multiple benefits over traditional methods, such as being non-destructive, non-invasive, and cost-effective. And because of that, it's been widely applied in different sectors, such as mining, agriculture, port monitoring, and oil and gas, where it can be used for identification of invasive species, endangered species, for assessment of environmental impacts, and so on and so forth. So once we know what's the research question we have, we collect environmental DNA from the relevant environmental substrate, which then get extracted in the laboratory. We then use gene-specific primers to amplify our target DNA, which we call amplicon. And next is library preparation to do the sequencing run. And the result of the sequencing is essentially lots and lots of amplicon sequences in forms of raw FASTQ files, which then we need to analyze. And for that, I initially set up a pipeline on my own local computer, just to do some of the routine tasks that is done for analysis of environmental DNA, which include some general quality filtering, assigning reads to sample, which we call it demultiplexing, some intermediate steps to change the format of the files to make it suitable for the main program, which does the denoising. And denoising is essentially trying to identify true biological sequences among reads. And then these unique denoised sequences are blasted against nucleotide database of GenBank. We also do a post-clustering curation step, which tries to remove erroneous sequences based on sequence similarity and also co-occurrence pattern. And finally, we use a custom Python script to assign those sequences to taxa that they belong to. This pipeline, however, had few challenges that I was aware of. To start, the large number of tools and the dependencies that had to be installed for the pipeline to work meant that it wasn't going to be easy to share it with others in our group, because then everyone else had to go through that pain of installing everything from the scratch. Also, the long-term maintenance of the pipeline was going to be challenging when those software and packages and also their underlying dependency would go through updates and changes. So eventually, something that is working beautifully today, in a year or 10 years time, or maybe even tomorrow, when I share the pipeline with someone who is going to run it on a different operating system distribution, then it wouldn't work anymore. And finally, the increasing amount of sequencing data that we get, it meant that the pipeline wasn't going to be scalable. I was lucky enough to attend one of the events held by Pawsey about containers. And a container is essentially a bundled package which contains all the runtime environment. That includes the application itself, the dependencies, libraries, binaries, everything that a package needs to run successfully. And containerization meant that we could monitor what version of the software we are using, and also to ignore differences in operating system distribution and the underlying infrastructure. So I was able to adapt my pipeline to use containers. And because of that, I was able to just set all the containers on Pawsey, and then just everyone else in our group could just run the script and use those containers. And I thought, that's great, and it couldn't have been better. But I was wrong, and I didn't know it until I attended another event held by Pawsey about Nextflow, a workflow manager which has some built-in support for containers and modules. And it also makes it very easy for a workflow to be deployed on various execution platforms. That could be your own local machine, cloud, or HPC. So again, I modified my pipeline, and it brought various benefits to that pipeline. For example, in my containerized pipeline, I wasn't able to say which of the process and which software is using more memory or it needs more time to be run. While Nextflow made it possible, so I could just tailor the computing resource parameters, because I know, for example, BLAST is more memory intensive than USearch, which is the free version of it, has some memory limitation. Also, another benefit was parallelization. In the old pipeline, one of the most time-consuming steps is demultiplexing, which the time is really dependent on how big our sequencing file is, how many samples we have, how many barcode files we have that needs to be demultiplexed. And in the containerized pipeline, it was just being done in a loop. First barcode file gets done, then next one, which was really time-consuming. But using Nextflow, it was very easy for me to parallelize that step. And just recently, I was able to do a complete run of the whole pipeline for 50 gigs of raw sequences, more than 160 samples, which was spread over 7 barcode files. It was all done in 1 day and 30 minutes on long queue, while with the old pipeline, it would have taken me at least 1 day just to demultiplex. The other important improvement is that it happens sometimes that the pipeline may crash, for example, because we may not specify enough runtime for it. And again, in the old version of the pipeline, we had to go back to the script and try to figure out which steps have been completed successfully and just comment them out or remove them to just resume the rest of the work. But Nextflow has this automated caching mechanism, which basically means it can skip the steps which have been finished successfully, and it just uses the cached result to run everything smoothly after you resume the work. Last but not least, portability is very important for us because not all the time we have large number of samples, so we do not need to run it on Pawsey. And also sometimes we actually even cannot do it because there are times that, for example, my colleagues, they go on a field trip, they collect the samples, they even have the facilities to do the lab workflow part, and they do the sequencing, they get the results, and they need to just run the analysis, but they don't have access to a stable internet connection. Nextflow made it very easy to basically to be able easily set up the whole pipeline on a local machine, because all the changes could be set up as variables in a small config file. And then that config files helps when you're running it with a flag of, for example, local, it can just pull all the containers, set up all the dependencies, and you can run it easily, which of course is going to take longer on local machine, but well, that's okay, I guess, sometimes. And to conclude, I just want to remind you of the benefits that all these changes brought to my initial pipeline. So now our pipeline is reproducible, it's scalable. Now we are able to use the computing resources more efficiently. It runs faster, it resumes easily, and it is portable. And with this, I'd like to thank everyone in our team at Trend and also all the collaborators for different ways that they contributed to this research. And my special thanks goes to Pawsey, not just because we are using their resources and because of the wonderful event that they host, but because their support doesn't end there. And a proof of that is a recently submitted manuscript describing the same pipeline in collaboration with Pawsey. And in particular, thanks to Audrey and Marco for all your advice and debugging and everything. Thank you.
Thanks, Mahsa, for this great presentation. So let us move on to the next speaker with Paul Hancock. So Paul is a research fellow at Curtin University, where he researches and teaches in the areas of astronomy and astrophysics. His main research interests include explosive transient events and radio variability, space situational awareness with the Murchison Wide Field Array, and software that supports and enables astronomy research. By developing new tools to automate data collection and processing, source finding and data analysis, Paul's goal is to better understand the information that we get from past, current, and future instruments, so that we can more reliably detect the rare and interesting objects that lead to new knowledge in the field of astronomy. So Paul today will be presenting a talk titled Porting Workflows to an HPC Environment. Thanks, Paul.
Thank you, Marco. And thank you, Mahsa, for giving such an excellent introductory talk. I feel like I can skip through a lot of the basics about Nextflow and containerization. So my project is about porting workflows to an HPC environment. And I've got the little subtitle here as Robbie to Robbie Next, and I'll explain what that's all about in a moment. So the basis of what I'm going to be talking about today, the types of data that we're working with here, is information that we gather from our radio telescopes. So what you can see on the slide here is on the right-hand side, there's a picture of a small piece of our radio telescope. That's a bunch of wire mesh on the ground with these fairly simplistic little dipole antennas. And when you combine all of the signals from this grid of antennas here, plus another 127 such grids, when you combine all of those together in a supercomputer, such as the Galaxy supercomputer at Pawsey, we can actually reconstruct what the sky looks like with radio light. So the image on the right is meant to depict what our eyes would see if we were sensitive to optical light, but also to radio light as well. So you can see in this image, there's that great big flame heading up towards the right, that's actually our galactic plane. That is hot gas and stars within our galaxy, giving off radio emission. Every other little dot that you see in this image is not a star. Every little dot that you see in this image is actually another galaxy, much like our own, with lots of stars and gas and dust, and also in particular a black hole at the center of that galaxy, which is eating a lot of gas and giving off a lot of radio emission. So at least with the low frequency radio astronomy that we do with the Murchison Wide Field Array, primarily what we're looking at is galaxies outside of our own galaxy, or we're looking at black holes, neutron stars, and regular stars within our own galaxy. So we use these simple instruments as depicted, and we use not simple computers to do all of our processing to create these images. And what we do with those images, is projects such as my own, which is to look at the changing radio sky. So the previous image was just like a constant map of the entire radio sky as seen from the MWA. What we're seeing up on the right-hand side here is a false color image, which is showing just one snapshot of the sky. The red dot dashed line is showing us the field of view that we can reasonably detect objects. The little yellow circle there is actually the Orion Nebula. That's what the Orion Nebula looks like with the radio telescope. Not particularly interesting. But what we're doing is we're taking lots and lots of different pictures of the same area of sky. And each of these snapshots we refer to as an epoch. So we've got lots of observations of the same part of the sky at different times. And for each of the stars, or rather each of the galaxies that we're seeing within these images, we want to build up a light curve. And so that's what's shown on the bottom right here. This is a light curve, which is essentially the vertical axis. It says flux density, but this is essentially the brightness of these objects. And the horizontal axis is how they are changing with time. So what's shown here is actually some simulated data. This is not a real observation, but this is simulated data about what a variable object would look like. So this is what a black hole would look like, for example. So the types of things that we're looking for are black holes that are feeding on galactic material, neutron stars or stars that are exploding in supernova, or potentially even neutron stars, which are these highly magnetized, very fast rotating neutron stars called pulsars. So these are some of the interesting objects that we want to see. And the method that I describe here is fairly simple. You make lots of observations, you make lots of images. For each image, you extract a list of all of the objects that you can see. You match those objects between different epochs. You build these light curves, and then you look for changes in the light curve. So that's the basic description of how it works. And in 2019, well, actually it was in 2018 when I did the work, I developed a software pipeline that automated this process. So this is the Robbie, the space detective, as I like to call it, is my pipeline. And this basic pipeline took 25 epochs of observations, because that's what I had available for my project at the time. It did all of the processing, which is sort of displayed here in this flowchart on the right-hand side. This is kind of my basic workflow of what I wanted my program to execute. All of this was executed on my own desktop computer. And I thought I was pretty smart because I managed to automate this process using Make. So Make is a program typically used for compiling C and Fortran code, but I kind of used it counter purposes to be able to implement the workflow that I've shown here. And it worked just fine. It worked well enough to be able to get useful scientific results, and for me to be able to publish my data, my methods, my analysis, and results as well. So that's this little paper that I've got this little green check mark next to here. So this was good enough to work on this fairly small project. Now, of course, there are many other people here at the Curtin Institute for Radio Astronomy who are doing similar projects using the same telescope. They have similar ideas. They're just pointing at different parts of the sky to be able to look for variable and transient radio sources. And so some of these people said, hey, I should use your software solution to do my analysis, save myself a bunch of work. I said, I think that's an excellent idea. And so I teamed up with Dr. Gemma Anderson, who likes to look at gamma ray bursts. So gamma ray bursts are a type of supernova. When the star gets to the end of its life, it explodes, lots of light goes everywhere. But the peculiar thing about GRBs is that they have this bright beam of gamma rays that are shot out in two directions. And sometimes that beam is pointed towards us here at the Earth. There's a satellite in space, which is monitoring the whole sky to look for these gamma ray bursts. And that satellite gives out automated messages every time it detects one of these objects. In a separate piece of work, Gemma, myself, and some others from Kyra had put a system in place so that the Murchison Wide Field Array could automatically respond to these events as they occurred. So without any human intervention, the telescope automatically points and begins observations of these gamma ray bursts. So the resulting data from a single gamma ray burst is 3,315 epochs of observations. So this is about half an hour worth of data, and we are making an observation approximately every half a second. The numbers don't quite add up because there's a few time steps that get dropped, but that's how it is. So we've got 3.3 thousand epochs of observations. So I thought, sure, why not? We can run this through my processing pipeline. But pretty quickly we realized that wasn't going to be possible because on a desktop computer, it would take about 6 months to do all of that processing. And that's 6 months full time if nothing breaks and if we don't use our computer for anything else. So I thought, well, ideally I'd like to run this on a supercomputer, to run this on Pawsey, but there are some other problems with the code. One of them is that Make is not actually designed to do what I was making it do. It breaks fairly easily. The parallel code that I had written is not managed efficiently by Make because Make doesn't understand which parts of the code use different numbers of cores. It didn't work on Gemma's computer, and it did work on my computer, so that was a bit annoying. And as I said, it didn't work on HPC systems as well. So essentially, oh yeah, also it was very hard to debug and fix. This little code snippet on the right here was something that I wrote about a year ago. And at the time of writing it, I knew exactly what it did. But today, I have absolutely no idea what this code is supposed to do. So when it breaks, it's essentially permanently broken. So of course, to overcome all of these problems, we decided to use Nextflow. And our decision to use Nextflow was mostly due to the fact that Pawsey was providing some training. So we thought this was a great opportunity. So we created ourselves a team called Robbie Next, as in the next generation, but also using Nextflow. And the 4 people on our team were Gemma and myself as the GRB people. We also had Nick and Keegan come along, not because they're involved in the GRB project, but because they were interested in using the same technology for some of their pulsar research work that they're doing. So there was 4 of us together. We got around for 4 days to go to the training and the hackathon. We had experts from Nextflow and Pawsey there to help us out. And within those 4 days, we were able to have a prototype of Robbie Next, which didn't actually execute the entire pipeline, but we could understand how the pipeline was going to work. So it didn't process the data, it just did little touch and echo of commands. So essentially it produced empty output files, but at least we could see that the program was going to work. We also identified all the components that needed to be replaced and upgraded. And there was lots of lots of different things that we were doing that just didn't work when you tried to run it on 3,315 files.
Hey, Paul, just letting you know that you have a minute left.
Okay, excellent, not a worry. So the Nextflow training was extremely useful. The additional work afterwards was to actually fill in the workflow, build Docker and Singularity containers to get everything working. And it's in this additional work phase that we got a lot of really crucial help from Pawsey and Nextflow people, mostly via Slack actually. It was extremely useful to be able to say, hey Marco, my thing doesn't work on Zeus properly, can you give me a little bit of help? And short turnaround, we got the help we needed and we had everything up and running again. So after converting everything to work on Nextflow, we now have a GRB project which is fully processed. We've actually processed the full data multiple times now. The paper, which I've just got a little snippet here, is in review with co-authors and will be submitted soon. We've got 2 more PhD projects which are using Pawsey, which using Robbie on Pawsey. And we're planning future projects as well. So this has been extremely successful for us. But also it means that now that we have people at Kyra who have some experience with Nextflow, we can start to teach some of the other researchers here, give a bit of an introduction, and then when we need more professional help, we can refer them on to the Pawsey experts. And the last thing I'd like to say is also we've now started to include Nextflow and a lot of the lessons that I have learned through this project in an honours level course that we're teaching at Curtin. So the benefits have been to us directly, to our colleagues indirectly, but also to our students as well. So I'll leave it there.
Thanks, Paul, for your very interesting presentation. So we're now moving to the last speaker for the day, Dr. Monica Kehoe from Deep Earth. So Monica is a plant virologist and molecular plant pathologist working for the Western Australian Department of Primary Industries and Regional Development. So she works in the diagnostic and laboratory services section, and the current work focuses mainly on the development, validation, and use of molecular methods for plant disease diagnostics across a broad range of broadacre and horticultural crops. Monica will be presenting today on the topic of incorporating supercomputing resources into diagnostic workflows. Up to you, Monica.
Yeah, thanks, Marco. Yeah, so hi everyone. Thanks for having me along this afternoon. So I'm just going to pull up my screen, and thanks to the previous 2 speakers as well for their great talks and some really nice introductions to some of what I'll be talking about as well. As Marco mentioned, I work in the diagnostic laboratories at the Department of Primary Industries and Regional Development as a molecular plant pathologist. And before that, I was a virologist. So all of the examples I talk about are to do with virology. So just a little bit of a background about what we do and why we do it. It's really important for us to know which pathogen or pest that we're dealing with in the samples and the crops that we look at. The main reason this is that a lot of these diseases can look quite similar, although they can look like they're having the same effect on the plant, they can be spread by lots of different methods, whether it's aphids, whiteflies, some of them are contact transmitted, some come in the seed. And so it's really important to make sure that we ask the right question, that we're using the right tools for diagnostics. So we use a really broad range in the laboratory, from serology right through to high throughput sequencing methods. And then sometimes we'll take it through to some phylogenetics. It's important to understand all of these things because depending on which insect or how the virus is being vectored will really dictate how you go about managing the disease that you've got. And that's not just relevant just to viruses, that can also be relevant to the bacteria and some fungi as well. So until a couple of years ago, we really only used high throughput sequencing as a research tool. we used to send all of our samples away to a third party for sequencing. We'd wait a while for the results. And it wasn't responsive enough to be part of a diagnostic workflow. So my PhD was my first foray into this type of work, looking at lupins here in Western Australia and a particular disease called black pod syndrome. And that was my first attempt at anything like this. And it took probably 18 months from sending the samples away to really getting a finalized data set. So it was really cool and it was a lot of fun, but the turnaround time needed to be better. So now we're really fortunate that we have in-house access at DPIRD laboratories to multiple sequencing technologies. So now our problem isn't getting the data, it's analysing it in a meaningful way that can really, I suppose that's the bottleneck now, is an efficient way to do the analysis. And more often, we're facing an increasing need to be able to rapidly diagnose unknowns. So I think often things of biosecurity concern that we may not have a specific test for already, and that we might have to wait a week or so for new primers or a new test to arrive in the labs before we can even validate that for our conditions. And so the other use, which is kind of what we were using high throughput sequencing before for, is to monitor existing pathogens and viruses and bacteria and fungi that we already know that are present in Western Australia, but we want to make sure that we're really across what's locally important and that new strains and variants aren't emerging or being introduced, and so that we can make sure that our diagnostic tools are as accurate as they can be. So the first step for us really was a Pawsey Uptake Project in 2018. So myself and Sam Hare, my colleague at DPIRD, we successfully got one going with the help of Marco and Yathu. And for that project, we really wanted to take that workflow for the Illumina data that we had been using on our own computers at DPIRD, using commercially available programs like CellC Genomics and Genius and a couple of others. So a local BLAST database we set up and Megan, but it was really time intensive, and it was one sample, one step at a time. Sort of manual curation every step, and it could really be weeks of time, and it didn't really fit into the workflow of what we did. So often I would start off the import of the data or the quality control, and then I'd get distracted by something else in the lab, have to come back and then realize that I hadn't kept going. So with the new workflow, it was really nice. Marco did a lot of really good work helping us to identify the open source software that we needed and to have the correct parameters. We did a direct comparison of results of a data set from our that we'd already processed using our old workflow and this new one, and we're really pleased that we could run all samples pretty much to first pass within 12 hours, and that's about 20 samples that previously probably would have taken me months to get through. And the other thing that's really nice about this is that it's reproducible, and we can also make it available. So it's available on the Pawsey GitHub at the moment, and we're still using this workflow as I speak right now. So one of the things I really wanted to demonstrate today was how we actually use this, I suppose, in the real world, and the types of applications that we're using this for. So one of these is grapevine leaf roll viruses in grapevines. So we have some pretty nice vineyards producing some pretty nice drops here in Western Australia. We're lucky, but there are there's a family of viruses, grapevine leaf roll viruses. There's about 6 or 7 of them that cause these types of rolling symptoms. And grapevine leaf roll virus 1 and 3 are probably some of the most important for the growers here. And it's common that there'll be mixed infections, but they can cause yield losses of up to 70% and reduce the fruit quality. So it's a really big problem for them. So the first project that I had was looking to fill a gap on virus diversity. So there weren't actually very many whole genomes of these viruses available. Worldwide, there are about 6 of grapevine leaf roll virus 1 and about 20 or 30 of grapevine leaf roll virus 3. And of all of that, there was just one single lonely grapevine leaf roll virus 3 genome from Australia. So we had no idea if the tools that we're using in the laboratory were really the most appropriate for what might be circulating in our vineyards. And then we wanted to use the data that we got from this project to design a new molecular test. And then we also used that new molecular test in a second project that was actually looking at the effect of these viruses on the end result of the wine. So I mentioned we wanted to make use of these new genomes to sort of, I suppose, to check that our molecular tests are the best that we could have. The coloured sequences, the coloured lines represent new sequences in this study. And this is grapevine leaf roll virus 3, and you can see that they're actually quite diverse. They look like a couple of little different subgroups there. And it turned out in the course of this work, we discovered that one of our sets of primers really it had the potential to miss one or two of these viruses within grapevine leaf roll virus 3. So we've already adjusted what we're doing in the lab, and now we're quite confident that we're picking up all of these known variants that we have in Western Australia, which is a good result for industry, because each year a lot of people screen for these viruses. And so we were able to add 25 new genomes, 12 of leaf roll 1 and 13 of leaf roll 3 to the data set, and it's a manuscript that's in preparation at the moment. But really the key was that this data was basically analysed in a day instead of probably months by the time we sort of dropped in and out on analysing it. And so from there, we took that data and we used it to design a new in-field test. So it was still a LAMP PCR designed against the sequences that we got from this project, so we could be sure again that's actually relevant to the variants and strains of these viruses that were in our vineyards. And it's also a method that has a really quick turnaround time. So in 30 minutes to an hour, you can get a result. And this will work in the field. You can crush up a leaf and really quickly use that as a template straight into the mix, get a result on the spot. But we do also use this as a tool in the laboratory to get quicker results sometimes, both which has had quite a lot of interest from growers and from agronomists who are considering maybe using this sort of tool for their clients. And then following on from that, we then took that new tool that we designed and we used it again directly in the field to screen for different vines that did and or didn't have the virus. And with the help of colleagues at Bunbury, Richard Fennessy, who's a really good winemaker, we actually got to make some wine and made different batches of it with and without the virus, and had some experts taste it. It was just I suppose it was a little bit of a pilot study. Some growers think that having the virus actually makes the wine taste better, but that really has flow-on effects for how they manage disease in the vineyard. So the results of this were promising. At least one of our tasters could reliably taste the difference between the wines. So we're hoping to get some funding to continue this work in the future. And so from here, where do we go now? Well, the Next Flow workshop at the end of last year, for me, was a bit of a game changer. I think a penny dropped for me. I've always said that I'm a biologist who dabbles in the computational, but I think I really started to understand a whole lot more after that workshop. And so we're now working on a similar pipeline for nanopore data, and we have one that's working quite nicely for small data sets. And we're also looking to sort of transfer that Illumina pipeline into the Next Flow, into a Next Flow pipeline as well. For me, this is really the next step for us in reproducible diagnostic tools for these kind of applications. It means that we can reproduce the results, we can apply the same settings to multiple samples. It makes reporting easier for us. And this is definitely an area where I suppose the technology and the tools are kind of ahead of the policy a little bit. And so in trying to make it as straightforward as possible, then it means, well, maybe we can have some say maybe in setting those standards, and having discussions around that policy, I think, is really important, and how this data and the results from it are treated. All of this really has been possible thanks to our involvement in the original Uptake project, and I'm happy to talk later on if anyone has any questions about any of the specifics of what we've done. But this work doesn't happen in isolation, and there's a lot of people to thank. My colleagues at DPIRD were all involved in the wine projects and in the survey for that. Industry as well have been very supportive, so particularly Voyager Estate and Allied Wines, who let us loose in their vineyards, and the staff at Pawsey, Marco, Audrey, Yathu helped us with the visualization steps a lot as well. And thank you.
Cool. Okay, so we might get started on a few questions that that we have for the panel. Through your uptake projects, what was the level of assistance that you've received, and how was your confidence before and after you received assistance from Pawsey staff? Anyone?
Is there a particular order?
No, please feel free to jump in.
Okay, so I guess before the uptake project, I was confident in programming and creating code that could run on a supercomputer, but my biggest headache had been orchestrating workflows, and that was something that I didn't have much confidence in at all. And the Nextflow workshop and then the follow-up assistance that was provided has completely changed that. And so now I think people in my office are kind of sick of hearing about Nextflow because I talk about it all the time and how useful it is. But it has really changed the way that I do work and the way that I plan projects. And essentially it means that I can engage in much larger scope projects than I thought possible before.
Yeah, so for me, when I started, I suppose for a start with the uptake project, I wasn't that confident. I'd managed to get things to run before and mostly using other people's scripts. And sort of I had enough knowledge really to tinker with those a little bit, but definitely through the process of the uptake project, I learned a lot, and Marco gave us a lot of support, and definitely built my confidence. But the Nextflow workshop really changed it for me. I actually attempted to build my own workflows now, and with a little bit of help to get them going from Marco and from Audrey, it's been really good. So I'm keen to do more, and I've been participating in the container workshops. So yeah, definitely keen to keep trying to build more and keep going. And I think a bit like Paul, people are a bit sick of me going on about Nextflow, but you know, it's such a great tool, and I think it makes it a bit easier for someone who's a bit new to this sort of thing as well. You can kind of compartmentalize bits of it, and that can be really helpful at times.
And what I really liked about it is the fact that I think on my own, I wouldn't want to go through just learning it just by myself, simply because I just didn't have like this concentrated time that I just sit and put that time to learn and then apply it to what I had. Because, well, if it wasn't for the actual hackathon and the workshop, I perhaps wouldn't go through all those changes, because in my brain it was, oh, okay, it's working, everyone can use it. And I didn't have the time to just go by myself and figure things out and improve it. So yeah, that workshop really, really helped.
Thanks, Mahsa. So I will keep on encouraging anyone that has questions just to post them on the chat, and we will pick them up. Meanwhile, I think there is an interesting follow-up then. So Mahsa mentioned the role of having a couple of days, a week off, just to focus on a single project. So, which is kind of one of the essences of hackathon events. So I'd be curious to hear by Paul and Monica what they're taking on this aspect as well. So the impact of having a week off just to focus on a research project.
Yeah, I agree. Like that was actually one of the keys to that week, I think, was that, you know, we stepped away from the office and the lab and actually focused on it in a really great and supportive environment. It was that was really key. So I mentioned that I've been participating in the container workshops, and that's been great that they've been online and accessible, but I suppose it was a little bit too easy there on the third one for me. Got a bit distracted enough to catch up with that one. Thankfully it's recorded, but you know, so having to set aside that time and go somewhere, it's not always possible, but when you can, it's I think absolutely worthwhile.
Yeah, I think for us, the timing in November was particularly good because it was after all of our teaching other commitments had finished, so we could actually dedicate those 4 days to the task. The other thing that I really enjoyed was not just the mentors who were at the workshop, but also all of the other peers. So all of the other groups who were at the workshop, because we were continuously encouraged to present on what we're doing and what our goals are, we could identify other groups who maybe had similar problems. And so we could go and ask them how they'd solved a particular issue. So there was more of that peer learning that went on when you're all in the same room, all with that same goal. Whereas doing something similar online, even if there's a chat forum for it, it's really easy to get quite disconnected. So the hackathon workshop combination was particularly useful in that regard.
Yeah, thanks, Paul, for raising this aspect of community building in a way, right, during the hackathon event. And now I have possibly a question for my colleague Chris as well, because this is not the first hackathon event that we have organized here at Pawsey to raise uptake and provide assistance to researchers. So maybe Chris, you want to comment on previous hackathon events?
Yeah, so we certainly have had a history of having a lot of hackathon events around GPU programming, which my colleague Pawsey, who's here as well, has run over the last couple of years. And they've been a great opportunity for users to sort of get up to speed with using some of the new accelerator technology and make great gains with making their code run faster. And I think it's certainly with this latest round of Nextflow training and other events that we've been doing, I think we've really recognized the usefulness of having that time following training to really apply it to researcher workflows. And it's something that we're looking to do as we start to roll out our developer training online, looking at things like OpenMP and MPI to do parallel programming, to sort of have those extra couple of days afterwards to really look at how to apply those to users' workflows as well.
Thanks. And just on that, we've talked about the Nextflow hackathons as well as a couple of the other hackathons that we've run. For Paul, Monica, and Mahsa, the format itself, where you were trained and then followed by the hacky days, would you be interested in that kind of format in the future? Was that helpful, or would you kind of change that around a little bit?
Yeah, I liked it. It was really useful to do some learning and then try to apply it.
Yeah, I definitely would recommend doing that same format again.
I agree.
Nice. That's good to hear. Okay, we have another question from our audience. They ask, are the Nextflow pipelines developed by you three presenters mostly processing data on HPC, or are they cloud resources? Are you using cloud resources?
Yeah, mine is all on Pawsey, mostly on Zeus, these ones. So everything I presented today is on Zeus.
So my stuff is also on Zeus, but I actually tried like a small version on my Nimbus account on the cloud. But the reason I'm not using it in particular there is just because I don't want to go through just downloading that whole nucleotide database and then looking after it and just, you know, update it on a regular basis, because I know that's done with Pawsey does that. So yeah.
Yeah, our workflow is being run on Magnus, on Zeus, also on some of the test cluster Topaz, and we're testing it now on the new MWA cluster Garawala. We've also been running it on desktop machines. So there's a few people at Cairo who have particularly small data sets, and for them it's easiest just to run that on their desktop machine. One of the main reasons that we're not using cloud infrastructure at the moment is similar to what Mahsa said, that just getting the data in and out of the cloud is not, well, I mean, it's probably not hard, but it's just not something that I know how to do easily. The MWA archive is hosted by Pawsey. So for us, Pawsey is really the right place to process the data. anything like a Google or Amazon or something like that, it's probably going to, spend most of our time transferring data, most of our money transferring data rather than actually processing it. So we haven't looked into cloud seriously yet.
Yeah, thanks for your comments on this question. There was just thinking back at what we discussed on the community aspect of the hackathon. So I was wondering whether you could also compare that to the contribution of the mentors. I mean, because it was there, so it's curious to hear what was the interplay between these aspects, right? Was that more important to have skilled mentors in place, or was it more important to be in a team and to talk to other teams, or maybe it's I mean, what was your feeling in those days in these regards?
I think all of them played an important role. I mean, it's nice to have that team aspect, but you're going to need the extra help from the mentors and everyone that's around is what really makes the day, really, I suppose.
Yeah, I wouldn't want to go to a hackathon that didn't have the mentors there. I think that was something that you couldn't do without. I think the team and community building aspect you could do without it, but it would be not nearly as good. So I would say it's not critical, but it's really, really useful. Mostly because I think there was at least one time when our team had a particular use case that we were trying to develop, or a particular thing that we were trying to do. And it was really good to have Paolo and Evan there to say, oh, actually we're developers of Nextflow, and what you're describing doesn't work, but it's a great idea, and we'll see if we can incorporate that into a future release of Nextflow. So that was also, I felt it was a really good way to help with the development of Nextflow.
Yeah, and what Paul and Monica already mentioned covers also my points. So yeah, it's a mixture of all of those points.
Cool. Mark, I don't know if you had any more to add on that, or should we go to the next question?
Yeah, we can. I think we can move on to other questions. So maybe we can ask Chris. We can move a bit more to broader topics related to this type of uptake events. And so Chris, are there upcoming opportunities for uptake projects in the next few months at Pawsey?
Yes, definitely. So for those will hopefully be aware, we're currently undergoing our capital refresh process of replacing Magnus and Galaxy and some of our infrastructure with a larger, bigger and better new system. And you can find out more information about that on our website. And with the arrival of that, there'll be a call for uptake projects. We did something similar with the arrival of Magnus, which was called the Petascale Pioneer Project, which allowed for some very high profile in-depth support for getting some codes up and running on those systems. And from what I've seen of what we're planning for the next system, it's going to be even better. So watch this space and keep an eye on the Pawsey news and events, because we're expecting an announcement about that soon. And it's going to be really exciting to be a part of. So keep an eye out.
And is there any limit, Chris, to how big an uptake project can be?
It really depends on the scope of the project and the availability of staff. So outside of the really big sort of early adopter projects with the arrival of a new system, we also periodically hold formal calls for uptake projects, and they undergo a sort of more rigorous review process. And they're sort of on a scale where you can have access to uptake staff to work in depth on a project for sort of 3 to 6 months for a substantial portion of their time. And then for smaller projects, it's a lot more informal. And so I think I talked about a little earlier around tickets of unusual size, where you just sort of come across opportunities for collaborations where it's, you know, a few days or a couple of weeks of work to do something. And that's something that sort of happens more organically.
Thanks, Chris. And maybe I'm just also commenting on this because this past months of unexpected working from home arrangements have also given us the opportunity to play with some remote formats for training and workshops with hands-on components and opportunities for people to apply learnings on their research problems. So right now we have been trialing this type of remote workshops for containers. We might be, as Chris mentioned, we are going to possibly expand this type of format to other topics such as advanced programmer trainings. But I was curious to hear actually about our three presenters today about any experiences they might have had recently in terms of remote forms of engagement, especially with regards to workshops, hands-on sessions. So I believe it's quite a hot topic and it can fit quite nicely in this type of context.
Yeah, well, I haven't seen, I suppose most of everything that I've attended in the last couple of months has been this kind of format. So a presentation and some discussion and chats and things. And I haven't had the opportunity to do anything that I suppose you'd call hands-on remotely like this, but you know, I'd give it a go.
Yeah, I also didn't have the chance of attending any hands-on virtual or remote type of workshop, but yeah, mainly meetings online.
Paul, any comments on this topic?
I think the only workshops that I've been to have been like a previous incarnation of this Pawsey Uptake project. So I think it was maybe 2 years ago now, there was a GPU hackathon that we did in Fremantle, which was similarly useful for a completely different project. And it was that same, you know, vendors who make the GPUs, people who make the programming libraries, and then a bunch of other teams that you could communicate with. We had that same very positive experience. As I said, I haven't done any of the online training yet, so I can't really comment on that.
Yeah, thank you. Chris, do you have any comments at all on this, or?
No, other than I think it's been really good to see the level of engagement that the community's had and continue to have in this time when it's been a lot harder to talk to each other. I think we've seen that there's a lot of people that are continuing to sort of come to these online events and online tutorials, and I think it's definitely a challenge. It's just more of a challenge to sort of have those more natural conversations, and that's why I think the Pawsey Ask Me Anything sessions on a Monday are a really good opportunity, because they are typically sort of a smaller forum. There's sort of one or two users that might come along and have a chat in a much more sort of informal way that's closer to those sort of discussions that we used to have in the breaks at training and the like. And so if you are looking for help with your workflows, that's certainly a great place to come along to.
Also, sorry, Marco.
No, nothing. I was just thinking whether any of our three presenters have any thoughts before we move on to the next part of this Pawsey Friday.
That's what I was going to ask as well.
I'm good.
I might have a quick question just to our presenters. Following on the topic, the previous topic around hands-on events. It sounds like you haven't taken part in any virtually yet. Was that from lack of anything that you're interested in being offered? or is that just hesitation or not really interested in something that would be a virtual hands-on type of event?
I think for me, it's just that I think we're extraordinarily lucky to have Pawsey in our backyard here in Perth. And so, you know, my preference is for those face-to-face events. It's, you know, the great people with great events, and you meet a lot of other researchers, you know, that you can learn a lot from in that kind of forum. And so I suppose most of what I've needed has been available in that format. And it's probably really COVID that's made people really think hard about those type of events. So I'm sure we'll see more of them moving forward, and I would be just as keen to participate in those if that was the only available option. So.
Okay, so if there was a topic that had a hands-on component, the topic you were interested in, you'd be open to that.
Yeah.
Okay.
Mahsa, Paul, any comments on these questions from Anne? Just wanted to mention the question was asked by Anne Backhouse, who is our training lead here at Pawsey, actually.
Yeah. Well, definitely, I would have been interested if there was something that I needed at a time. But I was personally mainly busy with some other online courses to learn how to make databases and other type of training, but I wasn't lucky to find a hands-on remote workshop about that at the time.
Yeah, same for me. Although instead of databases, for me, it was more about containerization. But yeah, for the most part, I sort of, you know, I'll do a hackathon, I'll apply that knowledge for a while, and then look for more opportunities. So even though it's been sort of 6-8 months since that hackathon, I'm still getting a lot out of it. And so I'm not in the mode of looking for new opportunities right now. But, you know, give it a few months' time. And yeah, whether that's in person or online, I don't have a strong prejudice one way or the other.
Okay, great. Thank you.
Okay, so do we have any more questions from the audience at all? We've still got about 5 minutes left of this Pawsey Friday event. Or would you like a chance to talk amongst yourselves, or any questions?
Oh, well, now I'm going to have to fill the space. So I'm curious. I'm curious. Sorry, I was dragged into some other discussions, but I'm curious what topics are of particular interest to individuals on the call. And I apologize if this was already asked.
No, it hasn't been asked yet.
Okay, great.
So if you guys like to, maybe you can comment in the chat. That way we can capture all of the topics that you might be interested in.
And indicate if they're if you'd like a hands-on component to those particular topics as well.
Yeah. Topics could be an area that you're struggling with, a point of confusion at the moment. It could be broader, how to use a system or a tool.
Because it's we find that it's been best practice, especially having worked with the Nextflow face-to-face and then having done the container ones virtually in a similar model, i.e. a learning component, then a guided hands-on, and then more of a kind of explore your own. That kind of model, kind of like a mini hack at the end, that kind of model seems to be very effective for big topics. And just curious if there are bigger topics like that, as well as if there are kind of smaller topics that are more of a, I would say just kind of the learning component.
So as Paul was very quick in putting in a comment, so I'm picking that up. So in the chat, Paul mentioned, yeah, we'll be interested in topics such as building and managing containers and how to orchestrate multiple containers together. So my
follow-up question then is, have you got an estimate of how, so if you think about the people, the collaborators in MWA and in your broader research team, how hot this topic is in the broader team there? I mean, do you reckon it is just you, or is that a kind of popular
Okay,
topic?
so it's a little bit weird because on the one hand, the MWA group is organized such that there's a few people, myself included, who act as like a bridge between the astronomers and researchers in the MWA collaboration using the computer and Pawsey, providing resources and support. So for us, it would be a case of there'd be about maybe 4 or 5 people who would be interested in knowing how to build and manage containers and provide those to the rest of our collaboration. And then everybody in the collaboration would need to know how to use those containers. Ideally, if we could set some aliases or wrapper scripts or something, I don't know. So on the one hand, the number of users being influenced is very large, but the number of people that would show up to that workshop would probably be 5.
Thanks, Paul. And as Aditi said, as Monica and Aditi said, please, anyone in the audience, feel free to +1 this topic that Paul raised. It's interesting for us to know, to have a feeling of the interest, because who knows, we might be offering this type of tailored events in the future. And actually, thanks to Anne for raising this topic further, because yeah, I believe it looks like these hackathon events are kind of a blend between training and actual uptake projects, right? You learn and then you apply straight after, and you see the benefit of what you just learned in your research after a week sometimes, or even less. So yeah, it really looks like a format worth playing with. Yeah, and then who knows, when we can all get back to the office, it might be face-to-face. Right now, yeah, possibly for the next few months, it will be most likely a remote event. But yeah, stay tuned with the Pawsey newsletter, let's say. And yeah.
Cool.
And,
okay, so it looks like we've kind of come to the end of the time for this event. So I just wanted to thank you all for joining us. We had a really good turnout today, so it's been really great to see, as Chris and Marco mentioned before, the support that we're still having to run our events. It definitely makes my job easier as the person that organizes a lot of these. So yeah, this kind of concludes the formal proceedings. If you were interested in today's Pawsey Friday, you can visit our website or sign up as a Pawsey friend to keep in touch and to see all of the upcoming events and training opportunities that we have. We're in the middle of planning a lot of webinar series, as well as some other kind of training events online, which will also be posted to our YouTube channel. So if you miss anything, we'll hopefully be able to capture it and you can watch it later on and do some of the hands-on components yourself. In terms of some of the events that we mentioned today, every Monday at 10 a.m. we've got an Ask Me Anything. It's what Chris mentioned, where it's a really informal opportunity to come and speak to our staff, to ask us any questions that you may have, and just have a bit of one-on-one informal kind of time. So that's listed on our website as well, pawsey.org.au/events. You can register there. The only reason you need to register there is so that you get the Zoom details. Yeah, we also have a Pawsey user forum coming up where we seek your feedback on how you use our systems, your experience with our services, and that's on the 20th of July. And I see in the comments that we've had a request from Marche to run more Pawsey Fridays. So we actually do have a special Pawsey Friday coming up on the 31st of July. This one's going to focus on the COVID-19 research that's been undertaken both at Pawsey and at NCI. So we'll be run a little bit earlier in the day just to account for our friends over East. So if you're interested in that, please register. And that comes out from the accelerated access that we had granted to applicants who wanted to dedicate some supercomputing time just for COVID-19 research. yeah, and like I mentioned before, if there's anything you'd like to revisit from this Pawsey Friday, it's going to go up on our YouTube channel soon. And we also have a survey for you to complete if possible, so that we can keep delivering these sorts of events to you. But other than that, I would just like to thank our 3 guests for joining us and putting on a really great presentation, and also to Marco and Chris for helping out with today's Pawsey Friday. Thank you
Thank you, everyone, and have a great weekend.